So the early CAR T cell experiments, and this is really expanding field, have been done in acute lymphoblastic leukemia. And what happens is that the T cell is taken out of the patient's body, is engineered, and when it goes back in again, it's got this chimeric antigen receptor, which not only does it bind it to the tumor cell, but it helps it destroy the tumor cell. So the idea is that a normal T cell will sit alongside a cancer cell, a leukemia cell, and won't destroy it. But after making it into a CAR T cell, it can now recognize and kill the leukemia cell. So this is a new way of destroying cancers by making CAR T cells. Now, the first child who was successfully treated by this was a young 7-year-old child in the States who had ALL. And the way that the gene for this chimeric antigen receptor is introduced is by something called a retrovirus, which is a virus related to the HIV virus. To be clear, it's not the HIV virus, but this is a virus that can introduce this CAR T cell, this engineered protein, into cells and make CAR T cells. And this is the patient where she met Barack Obama whilst he was still president. Now, CAR T cell therapy is really expanding now, and the ALL studies were the first, that was the first sort of target which is being used, but it isn't clear how far this is going to develop, but we would imagine that this type of CAR T cell treatment is going to be available for many leukemias, lymphomas, myelomas, but also for solid tumors, well, for other cancers beyond this. And I think just to show how vital it is that, that new CAR T cells develop, I want to just talk about a case of a patient that we've had some dealings with in Oxford. And I'm just going to read what he put on his Facebook page in case you can't read this. So this was from an 18-year-old, and he lives in St. Albans, and he says he's an ordinary teenager, except he has leukemia. Unfortunately, the bad kind. And he says that these days, 90% of leukemia patients are cured, and that's probably true in the younger, in childhood ALL. Sadly, those figures are not as good in adult ALL. But anyway, this is what he put on his Facebook page. I'm in the 10%, meaning he wasn't cured by ordinary chemotherapy. Intense chemotherapy, a bone marrow transplant from his twin brother Justin, and other treatment have all failed to kill the leukemia. I've been told I've just a few months to live. I was also told a year ago my leukemia was incurable. That was until a few weeks ago. A specialist team in Singapore have a brand new treatment. It cured Oscar, a 5-year-old boy in Singapore, and I've been told I'm eligible for the same treatment. But the treatment is in Singapore. It costs £500,000. It sounds huge. How are we going to raise that? But as my mum and dad say, we just need 50,000 people to give £10 and we're there, or 5,000 to give £100. That's it. Please give me a chance to beat this cancer and live. So this was someone aware of a new type of CAR T cell against not a CT19, but against CD7, which had been developed in Singapore. And as you can imagine, when these new treatments become available, then people internationally are very desperate to receive the treatments. And this is the sort of treatment, new treatment, that we want to develop here in Oxford. And talk about the kindness of strangers. Within 1 week, this patient had raised £500,000 for leukemia treatment. Incidentally, as you can see from this news story from the BBC website, whilst all of this was going on and he was fundraising and having his chemotherapy, he did very well in his A-levels and he won a place at Oxford University. So we came across him here whilst he came initially to Oxford, although he's now in Singapore and he's having his CAR T cell treatment in Singapore. And I think it's a reminder to us how vital it is that we develop these new treatments, CAR T cell treatments and other types of cellular treatments for patients who are otherwise going to be incurable. So I've been talking about a treatment where a gene is introduced into a blood cell and it makes the blood cell into a CAR T cell so it can destroy leukemia. I'm now going to pass you over to James. James Davies is a close colleague who works in the Department of Haematology. And he is one of the UK experts on genome editing. So, James, I wonder whether you could pass over now, if you could start sharing your screen, and James is going to talk, but just to pass over to James, this is the recipe book I was showing the slide of, Feast. And to give you some idea of how big the genome is, if you had all there are 700,000 letters in this book, approximately. The human genome is so big that if you had lots and lots of recipe books put one on top of the other, it would be 185 meters high in length. So the human genome is absolutely massive. That's bigger than the telecom tower in London. It's 2 Big Bens, or a little bit more than 1 telecom tower. So basically, there's a lot of genome there. And James is going to talk about amazing techniques which are available now for making specific corrections within the human genome. Thank you, James.
Thanks, Andy. So I run a lab at Oxford University, and we specialize in trying to develop cellular therapies using this new technology called genome editing. And this has kind of been in the news relatively recently. It's a new technology that was first kind of discovered or worked out how it worked back in 2012 by 2 women, Jennifer Doudna and Emmanuelle Charpentier. And amazingly, they were awarded the Nobel Prize for Chemistry this year. And this technology has completely revolutionized molecular biology. And it's sort of a pivotal moment, actually, really, because for the first time, we as a species are actually able to alter our own genome. And that's a very sort of strange and powerful thing to be able to do. And I think the way that it works, I don't know if this is projecting perfectly, but it's that what they discovered was that there's this bacterial protein called CRISPR-Cas9. And effectively what you can do is program it with a little piece of RNA that spells out the letters of the genetic code. And it will then go and search through all of the text of the genome and find the specific place that you've programmed it to go to. And you can use it to make cuts, or you can use it to insert bits of sequence, or you can use it to change the sequence very specifically at a single base pair now. And as Andy was alluding to, the genome is very big. So there are 3 billion letters in the genome. This is a book of the printout of it. And it's about the same amount of text as in the entire English Wikipedia. And the text in this book is absolutely tiny, okay? And what we're talking about being able to do is changing one single letter out of the 3 billion from, say, from a T to an A. And the technology that we're particularly interested in using this technology to fix inherited disorders of the bone marrow. And what we're talking about here is things like sickle cell disease, which causes the blood cells to change shape in the circulation and gives people painful crises, and it affects them and limits their life expectancy, really. And also thalassemia, where the bone marrow is unable to produce hemoglobin properly. And these people are dependent on blood transfusions every few weeks throughout their entire life. And what the plan is to use this technology, or what we're able to do now that we haven't previously been able to do, is to remove the bone marrow stem cells from the patient, as we would normally do for a bone marrow transplant. We then can change the genetic code in those stem cells very specifically at the place that we choose to, and then reinfuse them. And then they repopulate the patient's blood and immune system, but with very, very specifically altered genetic code. And so the first person to undergo a bone marrow transplant with genome editing was a very brave woman, a 35-year-old from Nashville, Tennessee. And she had sickle cell disease, and she underwent the bone marrow transplant about 1.5 years ago now, and the results were first published about 1 year ago. And this technology, on first go was extremely successful. So this lady had 7 sickle cell crises a year before the study and is now free of sickle cell crisis. And she's got normal levels of blood cell production. And the way that this technique works is actually to reactivate one of the fetal genes. And I guess you just have to trust me, but this is a spectacular result that they've managed to get half of her blood produced off the fetal gene, which has got a normal sequence. And this is to say that this technology isn't, for the faint-hearted. She had several complications during the transplant, which are the kind of things that we see quite commonly during bone marrow transplants. And she took quite a long time for the new bone marrow to make blood cells again. So this is a relatively early days for this technology, I guess, is the point that I'm trying to make. And we won't know the full safety of using this kind of genome editing technology for many years, but this is extremely promising. And we in the lab have been developing the sort of next generation of technologies for correcting thalassemia and sickle cell disease. And we're able to convert a single base from an A to a G in 95% of the stem cells that we want to edit. And we can do it in primary patient samples, so samples that have come from patients with the disease. And what's spectacular about this is that you can do this in hundreds of millions of cells all at the same time. So you can change one base pair out of 3 billion in 100 million cells all at the same time. And so I think this technology is going to be revolutionary. And we're really excited about trying to bring this into the clinic in the next sort of few years. And I think, I guess we'll probably take questions at the end, Andy. Is that what you envisage?
That's right, James. Probably work best if the three of us talk and then we do questions after that.
I guess I'm probably going to leave it at that, but if anyone has any questions, I'd be very happy to answer them later on. And I guess we'll hand over to Ronjon at this point. So, Ron, are you there? So Ron has just joined us in Oxford as one of the, presumably have to stop sharing actually, has just joined us. He's a professor of hematology and he's an expert on CAR T cells, and he's helping us to set up the centre. So it'd be very useful to hear what he has to say.
So I hope you can all see my slides and you can hear me okay. Thank you, Andy and Katrina, for organising this. I'm a new face in Oxford. I worked for 15 years in London in the transplant and cellular therapy unit here, but I'm very happy to join James and Andy and their team in Oxford and to help with this new project, which we're introducing you today and seeking your feedback. So we're trying to create a new centre, which is the Oxford Advanced Cellular Therapies Centre, which will allow us, for the first time in Oxford and for the patients that we treat, to allow quicker access to really cutting-edge new therapies based upon gene and cell therapy. Just to give you an example of how things are rapidly changing, and some of you will know this, but from the very first trials of CAR T cells in patients, the very first trials done by universities in the US, to the point when the CAR Ts became an NHS-funded therapy was just over 4 years. So things are moving very, very rapidly. What we want to try and do in Oxford is really to begin to get the benefit of the fantastic science that takes place in Oxford University. There are huge numbers of scientists working across the various institutes in Oxford who have real skills, as you've heard from James, in terms of gene modification and a better understanding of how cells work and how they can be modified, And what we want to be able to do in our new centre is to take some of those great ideas and be able to get those into clinic early and to benefit patients who have diseases like cancer or bone marrow failure that are causing difficulty in their lives. And what we're trying to create in Oxford is what we call a unique selling product for Oxford, because Oxford is different to many other universities and other places in the world. It has almost certainly some of the best scientists in the world. And what we're trying to do is create the clinical infrastructure so that these scientists who create fantastic ideas, a bit like the ones that have been discussed by James in the last talk, so we can go from those ideas and move quite quickly to first in human studies of gene therapies or cell therapies, in which we can conduct those studies in Oxford. And what we think will also add to this unique selling product for Oxford and benefit our patients is that those first patients going through those studies can also provide a great deal of information that will benefit future patients. So by understanding how these treatments work in the first set of patients that might be treated, we can better understand how to improve the therapies even more for future patients. It's no easy task to create what we call the infrastructure to run these studies. The National Health Service is really set up to deliver surgery and medical therapies. It's not really yet set up to deliver cellular therapies, particularly cellular therapies which are studies where they're very early phase, and so the first in human studies. So we have to create a great deal of infrastructure that will allow us to deliver these studies for our patients. So going from a great idea in the university, we have to have specialists that can help us design the trials. We need to be able to have the manufacturing that allows us to insert a gene of interest into a virus, a modified virus that can be safely used to modify cells collected from the patients, which then can be issued as a living drug into a patient. And thereafter, we would need to monitor patients very, very carefully over weeks, months, and years. And also to understand what happened to the cells that we gave to the patient and what their impact was on their disease. And in this way, we hopefully can also help improve our understanding of why the treatments work or don't work as well as we want, and that will lead to new discoveries and allow us to go through this process again. I'm just going to give you two additional examples that we are currently working on in Oxford. Andy has talked about the CAR T cells. We are also already in a position for a blood cancer called acute myeloid leukemia, in a position where we're beginning to understand how acute myeloid leukemia is different from normal cells. And we can begin to design what we call receptors that allow modified immune cells to recognize the cancer cells. And the way we do this is, as Andy says, you collect cells from the elbow vein of a patient. The cells go into the lab, they're modified so that they have a new receptor on their surface that can recognize the cancer, and then they're activated in the laboratory and expanded, and then they're reinfused into the patient where they have their effect. Some of you may know that CAR T cells can have side effects, and that's one of the reasons why the NHS has been quite cautious in opening these treatments in every centre. They're now focused in the UK in 9 adult centres. And one of the reasons is that the very first designs of the CAR T therapies, that they are effective in many patients, but they also have a different pattern of side effects to the ones that we normally expect to see from some of the drugs that we've used before. And I'm giving you an example of what might happen to a patient. So shortly after infusion, usually about 3 to 4 days after infusion, patients can get a very high fever, shown in this fever chart here. And sometimes, not in all patients, patients can get a bit muddled, usually about 5 to 6 days after the infusion. And this is shown here, a patient where we've got examples of their handwriting from days 4, 5, and 6 after the infusion. And you can see around day 5 that the patient's become quite muddled and their handwriting has become less good than normal. And this is a sign of what we call neurotoxicity. It's a side effect where chemicals are produced by the activated cells that are being given to the patient. And those chemicals have got into the brain and caused a patient to be muddled for a while. Thankfully, we've now got very good ways of dealing with that. But what it does show is that these treatments do have unexpected side effects at times. So we also have to be very careful in setting up our trial structure and our monitoring of patients, so that if patients do have any anticipated side effects, that we are in a really good position to care for them and make sure that problems are corrected. But one example of way we could do that, and this is something we're working on at the moment, is if you imagine these are the cells that have been infused into the patient, and these ones shown in orange are these active, overactive cells that are producing too many chemicals, more than there are needed, then we could modify these cells to stop them from overproducing the wrong chemicals that make patients feel poorly. So that's another example of what we might do. We need to create the necessary expertise, equipment, and skills in order to evaluate what we do in the following way. So the way I would think about it is something called product, fate, and impact. This means when we create a new product, for example, a cell product where the genes have been modified, we need to be able to characterize that in very high level of detail for every single patient. We also need to know what the fate of those cells are after they've been given to the patient. And this requires us to do blood tests, or what sometimes we can call these sort of liquid biopsies, where we can analyze them in a great level of detail using a whole schema of tests that allow us to track how many cells are left in the patient and how they're functioning, how they're working. And lastly, we need to know how the product characteristics, how the cells survived and functioned in the patient afterwards, what is the effect upon the patient's disease, what's their impact. And this sort of approach is actually critical to these new therapies. And we think Oxford would be very well placed to help us deliver that. Lastly, Oxford wants to create a position where we exploit the best of our science for the benefit of our patients. That's absolutely critical. But we don't want to exist in isolation. We want the work that we do to be of benefit nationally and internationally. So one critical thing that we would need to do, and where patients will be really important, is to think about the designs that would allow more and more patients, not just in Oxford but elsewhere, to benefit from these therapies. And this is, when we try and think about these ideas, we're talking about what we call, talking about therapy acceleration. So we're trying to go from a good idea that allows us to treat a few patients to get into a position where we can treat more and more patients and more places. And this impact network, which I'm the medical director, runs, we have over 23 sites, of which we're currently investing in 11 sites, it's increased from 9 to 11. But there are other examples of this, and where we go from what we do in Oxford to what we would do nationally, so that not just our patients, but patients across the country could benefit. I'm happy to talk about that more later. And that's all I've got to say. So I'll hand back to Andy and Katrina. Thank you.
Thank you, Ronjon. We do have a few questions and comments in the chat already, but I wonder, before we move on, I wonder whether, Andy, you could just give us a sense of some of the practicalities of what the proposal is. So where is this centre likely to be, and what will be required in terms of facilities such as intensive care beds and laboratory facilities and that sort of thing, just so people can start to visualise where this might be happening.
Yeah, no, thanks, Katrina. So we've really framed talking about, I suppose, two main types of cells, but Ron's explained that this is going to be the beginning of a number of different treatments. In terms of what it's like to receive the treatments, then some of them, the actual infusion is often very, very safe. And that can be given in a sort of day unit type setting. But we do have to be prepared that a subgroup of patients, and as Ron alluded to with CAR T-cell treatment, some patients can become very unwell, and potentially they might need intensive care. We were proposing that the centre, from the patient's perspective, would be located at the Churchill Hospital. At the moment, I think it's fair to say that we've got an open mind as to whereabouts on the Churchill Hospital it will be. I think if you gave all of us involved in this a magic wand, we would have enough funds to build it extremely close to the Cancer and Haematology Centre, joining onto that. The reality is, however, we've not got a magic wand, and building is expensive and time consuming, and so we probably would, at least initially, have a first phase where we use existing real estate at the Churchill site, so buildings which are already up at the Churchill site, and then perhaps in the long run our vision would be to have it more integrated. If there are patients, however, who are having treatments where we think they may become unwell, or if they do become unwell, then they'll go to the existing Churchill Hospital Cancer and Haematology Centre building, and then that would obviously be where the patients would be looked after. So that part of it is already there. I think in terms of the building, there's two important components. One is where the patients are looked after, where the patients are seen, where the patients are followed up, where they go for their complications. So that's the patient aspect. And then also, as Ron's been explaining, for this technology to work, the laboratory is what we also really need, where the cells can be manipulated. And that has to be to a very high standard, where the regulatory authorities would be happy with the product that's produced, and that can then be put into patients. So there's the laboratory and the patient area. And I think ideally we would put both of those areas adjacent, but I think it's not essential to do that. You can move bags of cells from the lab to the patient area, and they don't have to be adjacent. But obviously, as we're planning something, that would be our vision.
Great, thank you.